Hello everyone. Welcome to the session of Sexual Reproduction in Flowering Plants NCERT exercise part 2. So today also we are going to discuss about the rest of the questions from the NCERT exercise from the chapter Sexual Reproduction in Flowering Plants. Disclaimer: the images, text, and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issue, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using the generative AI, which is totally experimental. So, to practice and master the core concepts covered in the NCERT exercise through the problem solving. So, this is the learning objective. And in this NCERT exercise, the questions are from the topic that is 1.3, 1.4, 1.2, and 1.5. So, these are the different topics from this chapter that are having the questions today. The first question is, what is triple fusion? The first question is, what is triple fusion and where and how does it takes place? Name the nuclei involved in the triple fusion. So three questions are here and this questions is from the section that is 1.3 double fertilization from the NCERT book. So three questions are there. What is triple fusion? Right? You have to tell about the triple fusion. Then where and how does it takes place? How this triple fusion takes place? And the next question is, name the nuclei involved in the triple fusion. Name the nuclei that is involved in the triple fusion. So let us see the answer of this question. What is triple fusion? Where and how does it takes place? Name the nuclei involved in the triple fusion, right? So this question is from section 1.3. Let us see the answer. Triple fusion is the fusion of the male gamete with the two polar nuclei inside the embryo sac of the angiosperm. So this is one of the very unique feature of the angiosperm. This triple fusion only occur in the case of angiosperm, right? So what happened in this? As you all know that how many male gametes are there? Two male gametes are produced. And out of these two, one male gamete is involved in the process of syngamy. One is involved in the process of syngamy. And the zygote is formed in that process. But the next male gamete, the next male gamete moves toward the center of the central cell. There is a central cell in the embryo sac, you all know. And in that central cell, how many polar nuclei are there? Two polar nuclei are there. Two polar nuclei, right? And that is the part of the embryo sac only. So the second male gamete moves towards the two polar nuclei in the embryo sac and the result is triple fusion. So the process of fusion takes place inside the embryo sac. Where this fusion is taking place? This fusion is taking place inside the embryo sac only. When the pollen grain falls on the stigma. So here we are going to study about how does it takes place. This is the second question. Where and how does it takes place, right? So when the pollen grain falls on the stigma, they germinate. The pollen grain germinate. So there are many, like you can say the germ pores are there, right? That germ pores give rise a pollen tube. And you all know this pollen tube is the extension of intine. That pollen tube is the extension of intine, right? So the pollen tube emerges from this germ pore, right? This germ pore, and it germinates. Then it passes, or you can say penetrate the stigma style and reaches to the ovary. So that gives rise to the pollen tube that passes through the style and enter into the ovule. So this pollen tube enters into the ovule. But there is some chemical reaction that is also taking place on this stigma. You all know that, right? The chemicals that is released by the stigma part, the hormone that is released by the stigma part helps the pollen grain to germinate. But if it is compatible one, if it is compatible one, if it is not compatible, then there will be no chemical dialogues between the pollen grain and the stigma, right? Self incompatibility phenomena I'm talking about. So that also occurring at the same time. But if it is a compatible one, then the stigma will release chemicals, hormones that are helpful for the pollen grain for the germination process, right? Okay. So the pollen grain germinate at the stigma. After this, pollen tube enters one of the synergid. So pollen tube makes its way from the stigma to the style, then it reaches to the ovary, and then it make a turn, and this pollen tube get enter inside the ovule. That enter inside the ovule through the micropyle. The micropyle is a pore-like structure, right? And through that, this pollen tube get enter inside the micropyle. Now, one of the synergid, now here it is written very carefully, one of the synergid allow the pollen tube to get enter inside it, right? The pollen, the synergid cell is having actually what? Filiform apparatus. The synergid cell is having the filiform apparatus. What is the function of filiform apparatus? Guide the pollen tube. It guide the pollen tube. It releases the chemicals that attracts the pollen tube guiding. So how to guide? The pollen tube entering inside this micropylar pore, and at the same time this filiform apparatus will release some chemicals. So that chemicals will attract the pollen tube toward itself. So it is guiding to get enter inside this synergid cell, right? Okay. Once it get enter inside the synergid cell, what will happen? Okay. The pollen tube will the pollen tube will rupture. Okay, the tip of the pollen tube will be ruptured with the help of the chemicals released by the filiform apparatus only. And all the chemical substances, means the cytoplasm, the gametes, the male gametes which are present in the pollen tube will be released in the synergid cells, right? So here it is written, after this the pollen tube enter one of the synergid and releases two male gametes there. It releases the two male gametes there, right? Out of the two male gamete, one gamete fuses with the nucleus. One gamete fuses with the nucleus, and that results in the formation of zygote. So the fusion of first male gamete with the egg cell, that will form the zygote, and this process is called as syngamy. Right? Now, I'll just want to tell you about this filiform apparatus. It is involved in the release of the chemicals that attract the pollen tube, guiding towards the micropylar end of the ovule. So this is also one of the function of the filiform apparatus, right? So if you want, you can write one particular line in between that. Otherwise, it is okay, because this is written here that one of the synergid is involved. Actually, the filiform apparatus is very, very important, like part of the synergid itself. Yes, of course, difference between the pen, endosperm, and endospermic tissue. I'll come and explain one by one, but let me first explain this question, then I will come to this question. The other male gamete, Now we are having one more male gamete, because here you have all, it is clearly mentioned there that the pollen tube is having the two male gametes. So one male gamete was involved in the process of syngamy. The second male gamete fuses with the two polar nuclei. Two polar nuclei. Where these two polar nuclei are present? In the central cell. In the central cell of the embryo sac, right? So other male gamete fuses with the two polar nuclei present in the central cell of the embryo sac, and it will form triploid primary endosperm nucleus. So you got that one particular answer of your question. PEN, primary endosperm nucleus. And this primary endosperm nucleus is what? This is the nucleus only. We are talking about nucleus only. PEN means primary endosperm nucleus. The nucleus that are going to involved in the process of endosperm formation. That is going to involved in the process of endosperm formation, right? So that nucleus is called as primary endosperm nucleus, and it is triploid in ploidy. Triploid in ploidy, how it become triploid? Because one male gamete, that is the haploid. That is the haploid. And the two polar nuclei, and both these two polar nuclei are also haploid. These two are also haploid. So three gametes, three nucleus that are haploid get fused together, and the ploidy become triploid. So primary endosperm nucleus is the nucleus that is involved in the process of endosperm formation, right? So here you can see in the diagram also, the male gamete is present inside this pollen tube. It is released in the synergid cell. Filiform apparatus is helping and guiding to get enter this pollen tube in the synergid cell, right? So the two male gamete released here. One male gamete makes its way toward the egg first of all, and it fuses with the egg. Syngamy occur, zygote is formed. Okay, this is first fertilization. Now the second, the polar nuclei, here you can see the two polar nuclei are fusing together, and the male nuclei, the second male nuclei is reached here. Later on, this two polar nuclei fuse together, and this male nuclei also fuses together. So triploid nucleus is formed, and that is called pen primary endosperm nucleus. Clear? Now endosperm. What is endosperm? Endosperm is, if it is written endosperm, then this triploid primary endosperm nucleus divide and form many nucleus, and later on the cell wall is laid and the total endosperm is formed. The total endosperm is formed. So the endosperm is made up of many cells and nucleus, right? If you're talking about the endospermic tissue means all the endosperm cells unite together and they form that the particular tissue, that is endospermic tissue. Endosperm, it once the pen will divide, it will form two nucleus. Then this two nucleus again divide, then form 4, 4 to 8, 8 to 16, 16 to 32, 64. In this way, the nucleus multiply and multiply. Free nuclear division. Initially, no cytokinesis is taking place. So that is the endosperm. That is the endosperm. But once the cytokinesis also takes place, the cell wall is laid around each nucleus, the endospermic tissue is formed. Right? So you understand pan, you understand endosperm, and you understand endospermic tissue now. The three different things are there, right? Okay, I'm moving forward for this, the rest part of this question. So since this process is involved the fusion of three haploid nuclei are fusing together, so it is called as triple fusion. Why it is called triple fusion? Because three haploid nucleus is formed, and that give rise the triple fusion. That process is called as triple fusion actually, right? And what we get at the result? Triploid primary endosperm nucleus. This is the nucleus, right? It results in the formation of endosperm. Now, this uh, pen primary endosperm nucleus, that will give rise the endosperm. one male gamete nucleus and the two polar nuclei are involved in the process of this triple fusion, right? So only one male gamete nucleus and two polar nuclei that are involved in this triple fusion. Now we are moving to our next question. Why do you think the zygote is dormant for some time in fertilized ovule? Okay, before moving to this question, I just want to there was a question asked. So, can you please explain the difference between the pen endosperm and endospermic nucleus? I explained this, so I hope it is clear for you. The pen is the primary endosperm nucleus, which is single nucleus. We are not talking about anything, only the nucleus, primary endosperm nucleus, and this is the result of the three haploid nucleus fusion. One is the male gamete and another is the two polar nuclei. So three nucleus fused together, and that is why its ploidy is triploid. Another thing, they are talking about the endosperm. Endosperm is what? This primary endosperm nucleus divide and make a large number of nucleus. No cytokinesis is taking place initially, right? But once the large number of nucleus is formed, so that stage is itself called as an endosperm. So I'll taking an example of this coconut, the coconut, tender coconut, the green one, right? So you drink that coconut water. So what is that? That is filled of all that nucleus, right? Endosperm nucleus. And later on, what happened? This water settles at the inside, and you have this white kernel thing, right? So that is actually the endosperm tissue, endospermic tissue, because each nucleus surrounds some sort of cytoplasm and cell wall is laid all around it. And all this endosperm forms transform into the cellular structure. So this whole endosperm is now cellular, and there are so many cells. So this whole tissue, now the group of cells are there, na? So that is tissue, and its function is to perform what? To provide nutrition to the developing embryo, right? So that is the endospermic tissue. Okay. Okay. Now I'm taking the second question. Why do you think the zygote is dormant for some time in a fertilized ovule? Very, very important question. Why do you think the zygote is ceased its growth, no division? It is in the dormant stage for some time. For some time. So why this is happening? This question is based on our NCERT section 1.3. Okay. So let us take this question. So why do you think the zygote is dormant for some time in a fertilized ovule? The zygote is formed by the fusion of the male gamete with the nucleus and the egg cell. Dormant. Okay. So dormant means all the metabolic activities is at the lowest level. At the lowest level. No division is taking place, no activity is taking place. Right? We have read about that zygote. When formed, it divide, it makes a number of cells, and the embryo is formed. But here, that it also happened here, but after some time. So why it is taking some time, and at that time what is happening in the zygote? So zygote ceases all its activity, stop all its activity, all the metabolic activities is at the lowest level, no division is taking place. So that is the dormant stage of the zygote. Okay, no activity is taking place here, only the diploid zygote is there. Diploid means it is having the result is the fusion of the two male, one male gamete and one egg, right? One female gamete and one male gamete. So that is the result of the syngamy, the zygote. No division taking place, no activity is there, right? So that is in the dormant stage. Now, what is happening then? The zygote is formed by the fusion of the male gamete with the nucleus of the egg cell. That we all know, right? This is the result of the syngamy. Zygote is the result of the syngamy in which one male gamete and one egg cell nucleus fuses together, right? Okay. Now coming to the reason why they are in the dormant stage. The zygote remain dormant for some time and wait for the endosperm to form. It is waiting for the endosperm to form. It means first syngamy is taking place and after that triple fusion is taking place. But this triple fusion is taking place for formation of endosperm. The endosperm will form earlier and the zygote that leads to the formation of embryo that will form after the endosperm formation takes place. Why so? Which develops from the primary endosperm cell resulting from the triple fusion, that all we know. But why this is happening, that zygote formation takes place earlier and endosperm formation takes place later on, but still the endosperm form first? It is because the developing embryo required a lot of nourishment. They required nourishment, they required nutrients, they required food. Yes, right. So where the food will come? So that zygote wait for some time so that the endosperm can be synthesized, the endosperm can be made, and that endosperm will provide a lot of nourishment for the development of the embryo, right? Yes, the zygote remain dormant for some time and wait for the endosperm to form, which develops from the primary endosperm cell resulting from the triple fusion. The endosperm provide food for the growing embryo. Yes, the endosperm provide food for the growing embryo. And after the formation of the endosperm, further the development of the embryo from the zygote starts. So that's why the zygote takes some time, be in a dormant state, and once the endosperm is formed, it become active, it divide and divide, and while dividing it required the food material, the nourishment, and that is provided by the endosperm. Okay, so very important question this was. Next question, differentiate between. So you have to differentiate between the four questions are there in the section wise. The first one, hypocotyle and epicotyle. You have to differentiate between the hypocotyle and epicotyle. Second, differentiate between coleoptile and coleorhiza, right? Third question, differentiate between integuments and testa. Integument and testa. Fourth question, differentiate between perisperm and pericarp. So 4 differences they have been asked in this particular question, and this question is based on the NCERT section 1.4, right? So let us see the answer of this question. So this was our question. So we will take one by one the differences. So the first difference is between the hypocotyle and epicotyle. Hypocotyle and epicotyle. The portion of the embryonal axis which lies below the cotyledon. There is a level of cotyledon, right? And they are taking in the dicot embryo. This you see in the dicot embryo. So the portion of the embryonal axis that lies below the cotyledon, in the dicot embryo, is known as hypocotyle. You have to remember this word, below the cotyledon, that is the hypocotyle. The region of the embryonal axis that is below the cotyledon in a dicot embryo is known as hypocotyle. Very simple. And what is the epicotyle? The portion of the embryonal axis which is lie above the cotyledon. Okay, the embryonal axis, you see the embryonal axis, and the part that is above the cotyledon on the embryonal axis, that is known as epicotyle in the dicot embryo. This is the first difference in between the hypocotyle and epicotyle. The next difference, it terminates with the radicle. This hypocotyle, where it end? It end where there is the termination. Where is the radical present? There is a part where the radical is present. So it start from the region of the cotyledon and end with the radical, where the radical is present. So that part in between the radical and the level of the cotyledon, below the level, so that is the hypocotyle, right? Now coming to this epicotyle, it terminates with the plumule. Epicotyle region terminate with the plumule, where the region of plumule is there, right? So that is the epicotyle region. Very, very important. Next difference. Differentiate between the coleoptile and coleorhiza. You have to differentiate between the coleoptile and coleorhiza. So it is a conical protective sheath-like covering. It is a sheath-like covering, and you will see the diagram then in the NCERT. Then the shape of this coleoptile is like cone shape, and that is why it is called as conical protective sheath-like covering that encloses the plumule. That encloses the plumule part. It is providing the covering to the plumule part. It is providing protection to the plumule part, but because it is a very delicate part, right? So it is covering this plumule part in a monocot seed, right? This you can see in the monocot seed. So coleoptile. Now what is the coleorhiza? It is an undifferentiated sheath. This is again a sheath-like structure that encloses the radical and the root cap. So it is covering, this covering is protecting the radical as well as the root cap in a monocot seed, So this is the difference between the coleoptile and coleorhiza. Now the difference between the integuments and testa. Integument and testa. It is the outermost covering of an ovule. You have seen the covering of the ovule, outer integuments and inner integuments, right? So that is the testa, right? And it provides protection to it. It is providing protection to the ovule, right? Now the testa. What is testa? So testa is the outermost covering of a seed. When this ovule transformed into seed, the ovule transformed into the seed after the fertilization process. Okay, when you see the seed is having a particular cover over it. So that covering is called as testa, right? Now this integuments, I've already told you, well, I was explaining the ovule part, right? The ovule arises from the chalazal end of the ovule, from where this integument start formation takes place. The formation of this integument takes place from the chalazal end only, right? And this testa, how this testa is formed? The question should be asked, right? How this testa is formed? Actually, it is derived from the outer integuments of the ovule after the fertilization. You have seen the integument, outer integuments and the inner integument, right? So the outer integument after fertilization transformed into testa, and that forms the covering of the seed. So it is actually the same thing. It is actually the integument and the testa is the same thing. The integument, that is when there is no fertilization took place in the ovule, right? So that time you see this ovule is covered by the integument, right? The ovule is having the covering integument. And when the fertilization takes place, the outer integument transformed into the testa, right? So it is a post, it means that testa is the post-fertilization structure, right? The testa is a post-fertilization structure and integument is the pre-fertilization structure. It is a pre-fertilization structure, right? So this is very, very important. Now I'll move to the next difference. I hope it is clear. Difference between the integument and testa. Okay, moving to the next difference. Differentiate between the perisperm and pericarp. Perisperm, it is a residual nucellus which persists. What is the perisperm? You all know that endosperm is required for the nourishment to the developing embryo, right? But all the embryo is not used. Sometime some part of the embryo is left over. So that part of the nucellus, this nucellus part is left over. So that part of nucellus is called as persistent nucellus, not the endosperm. Actually, this is the nucellus, right? This perisperm is talk about the persistent nucellus, not the endosperm. So it is present in some seeds such as beet and black pepper. Beet means sugar beet, and black pepper. They are having persistent nucellus. Actually, the nucellus is also involved in the process of providing nutrition to the developing embryo, and that all the nucellus cells are used up in that. But sometimes some part of the nucellus is left in the beet and black pepper. That residual nucellus, or you can say the leftover of the nucellus cells, is called as perisperm. Okay. Now, the similar word is pericarp, but this pericarp is totally different from the perisperm. It is asked because the difference between these two, because these two terms are similar, right? So, to avoid any confusion, you should be like know about these two terms, perisperm and pericarp. Pericarp, it is a ripened wall of a fruit. It is a ripened wall of a fruit. So how the fruit is formed? The fruit is formed from the ovary, right? It forms from the ovary. When the the ovule get fertilized, when the ovule get fertilized, the ovule changes to seed and ovary changes to the fruit, right? So when the fruit formation takes place, the wall of the fruit ripen, right, which develops from the wall of an ovary only. So that wall is called as pericarp in the fruit, right? So next question is here. The next question, why is apple called false fruit? Which part of the flower forms the fruit? Right. So this is again the question comes from the part of the NCERT, that is section 1.4. 1.4. Why is apple called false fruit? So this was a question. Let us see the answer. The answer of this question, why is apple called a fruit? Let us see that. An apple is called a false fruit or accessory fruit because the fleshy edible part develops primarily from the flower receptacles, that is the thalamus. So why they are calling this thalamus? If the fruit is fruit, why this true and false fruit term is coming out? So generally what happened, the ovary transforms into fruit because the ovule is present inside the ovary only, and after fertilization, whatever the extra food material that gets synthesized by the plant, it is transported to the ovary to get store that fruit in the fruit form, right? All the nutrients are transported to the ovary, and that slowly and slowly this ovary becomes the fruit. But in some fruits, what happens? This ovary is totally embedded inside the thalamus, like we see in the case of apple. The ovary is embedded inside the thalamus, that is the receptacle part, right? And the nutrients or the food material that is synthesized by the plant by the process of photosynthesis, the extra food material is transported to the flower, okay, to the flower, actually after the fertilization. So here the function of the ovary is taken by the thalamus, because this ovary is embedded inside the thalamus. So all the nutrients, all the food material gets stored in the thalamus itself. So here the thalamus is storing all the food material and it becomes the fruit, right? Instead of ovary, the thalamus. So let us see this small diagram I am drawing here. This is the diagram of an apple. The apple is like this, and you can see a cross section. If you cut it, it is like this, and it is having seed like here, right? So this is the thalamus, right? This is the ovary, and this is the seed, right? So this is the ovary. You see this ovary is totally embedded inside this thalamus part, right? So the food material which is transported here by the process of photosynthesis, so it gets stored in the thalamus instead of ovary. So such fruits are called as false fruit or the accessory. It means the true fruit is that fruit in which the ovary transformed into fruit. The ovary itself transformed into fruit, it stored the food material, right? Sir, so in apple, the thalamus wall becomes the pericarp. Yes, it is the pericarp. This is the fleshy part that is edible, right? Okay. Now coming to this, what we are discussing about. Yes, you have seen the strawberry also. The strawberry is also having this the thalamus in which all the ovaries, there are multiple ovaries that are embedded inside the thalamus of the uh, strawberry, right? So there also we see the formation of false fruit. The fruit derived from the ovary and other accessory floral parts are called false fruit, right? So if you derived the fruit derived from the ovary, then it is true fruit. But it is if any other floral parts, like here we see the thalamus, so that is the false fruit. On the contrary, the true fruits are those fruit which develops from the ovary only, but do not consist of the thalamus or any other floral parts, right? In an apple, the fleshy receptacle forms the main edible part. Hence, it is a false fruit. So, in the case of apple, the receptacle or the thalamus forms the main edible part. That is the edible part, not the ovary one, right? So, that is why it is called as false fruit. Next question is here. What is meant by emasculation, and when and where does a plant breeder employ this technique? Two questions are again here. What is meant by emasculation, and the second, when and why does a plant breeder employ this technique? This is coming from again the NCERT section 1.2. So let us see the answer of this question. Emasculation is the process of removing anthers from the bisexual flower without affecting the female reproductive part pistil, which is used in the various plant hybridization technique. So for to understand this emasculation, suppose you have taken a bisexual flower, and be ensure that this the pistil part of the flower is immature at that particular time. Okay, so as soon as the flower blooms out, you take that flower. With the help of forceps, you remove its male reproductive organ, that is the anther, or you can say the stamen. You can remove whole stamen part slowly and slowly without affecting the female reproductive part, that is the pistil. No damage should be there to the pistil part, right? So that flower is now called as is a female part only, so that becomes the female flower, right? So the emasculation is performed by the plant breeders in the bisexual flowers. Remember, bisexual flower. to obtain the desired variety of a plant by a crossing a particular plant with the desired pollen grain. Now, this term is coming here, desired pollen grain. We will understand why this term is here. So, first of all, emasculation. So, the removal of the male reproductive organ from the bisexual flower with the help of forceps is called as emasculation. That process is called as emasculation. So why we have taken this? Because we are already having some desired pollen grain. So one flower you have already taken in which you have done the process of emasculation. Now you have another flower that is from the desired plant, right? So you will collect the pollen grain from that flower, that plant flower, the desired one, right? So that actually you want that pollen grain to fall on the first flower which you have emasculated. So that's why you have removed all the stamens. So by chance it could happen that the pollen grain from the same flower could fall on that stigma. So to avoid that, you have taken out all that stamen, and this process is called as emasculation. Now after this, one more process is there, bagging. You will take a butter paper bag and you will cover it after the process of emasculation. I'm coming to this question. Okay, so this happens like this. You have emasculated the flower, cover it with the bag. Now you collect the pollen grain, the desired pollen grain. You remove the bag. And this time the stigma is mature. Now put all the desired pollen grain with the help of paintbrush on this stigma, and again cover it with the paper bag. So to remove the anthers from the flower and cover with the bag before they open. This ensures that the flower is pollinated by the pollen grains obtained from the desirable varieties only. So this we get ensure that pollination by the pollen grains, pollinated by the pollen grain obtained from the desired. This is the surety that only this pollen grain is coming to the stigma, right? So whatever the pollination is there, this is by the desired pollen varieties only. So later on, the mature, viable and the stored pollen grain are dusted on the bagged stigma by the breeders to allow the artificial pollination to take place and obtain the desired plant varieties. Okay, so your question is here. Right, sir, banana have no seed, then how we can grow? So here the answer is from this question also. If one can induce the parthenocarpy through the application of the growth substances, which fruit would you select to induce parthenocarpy and why? So this banana, actually you can produce the parthenocarpy food with the help of spraying some hormones like auxin. Now, in the case of banana itself produce the auxin. It itself produce the auxin so much that before fertilization, it forms the fruit. Naturally, it is happening naturally. So, banana without fertilization also, you are getting bananas, right? And you can see how many everywhere you can see banana in this particular season, right? The fruit vendors are selling so much bananas, right? So banana have no seed, then how we can grow? It can grow itself with the help of naturally. This auxin is produced in their body and it is transferred to the flower, and before fertilization it produces the banana. And one more thing, the banana reproduces asexually very fast, right? So this is the answer. But there must be one more question, then how the new varieties of banana is made, right? How the new varieties of banana is made, right? So by the process of Like you can do that, you can put some seeds, and with the help of tissue culture also, and in the tissue culture also technique, you can mix the genes from the two different varieties, right? Okay, so naturally this parthenocarpic fruit is made by this banana. Yes, by the suckers that the new plant comes out from the corm. Actually, there is a you can see the elephant foot, the suran, which we call in Hindi, right? It is the corm-like structure. That is the modification of the root, and that will give rise a new suckers in that corm in the case of banana. And this banana is what? It is a herb or tree. Is banana a herb or a tree or a shrub? What is it? Can anyone can tell me? It is a giant herb. Very good. It is a giant herb. It is a herb. It is not a tree, right? And banana is very, very important plant. It is providing so much useful. Not a single part of the banana is wasteful. You can see the leaves. The leaves of banana is used as a plate to serve the fruit in the South India, right? The fruit is involved in making the chips, making it is used as a fruit, it is making vegetables, right? And also the peel of the banana is also used in making vegetables, right? Another thing, coming to the flower is also used in making the vegetables, right? And the stem part of this giant herb is used in making clothes. Saris are made from this, the fiber of this banana herb. And the soft stem part is also used in making vegetables. So banana is very, very useful plant. Okay, now I'm taking this question which is on the screen. If one can induce parthenocarpy through the application of the growth substances, which fruit would you select to induce the parthenocarpy and why? This question is coming from the NCERT section 1.4. So let us take this question. So if one can induce parthenocarpy through the application of the growth substances, which fruit would you select to induce parthenocarpy and why? Parthenocarpy is the process of developing fruit without involving the process of fertilization. No fertilization is there or the seed formation, but still the fruit is formed. Therefore, the seedless varieties of economically important fruit such as oranges, lemon, watermelon, etc. are produced using this technique. There is a pollen pistil interaction. So if you pollen pistil interaction is there, yes, this is occurring naturally when the pollination, after the pollination, but you can take the two different cells of the two different varieties of the banana. I'm just taking the example of banana in the laboratory. Two cells, and you can fuse those two cells. Haploid cells you can take and you can fuse those two cells. And what happened then? The two nucleus fuses together and form the diploid cells. And later on, they could form, with the help of tissue culture, you can form that whole plantlet. Hmm? So this is pollen pistil interaction is there after in the pollination just to avoid the incompatible one. The incompatible one. But if you want, still you want that the fertilization could happen there, then you can go for this plant tissue culture technique, right? So therefore, the seedless varieties of the economically important fruits such as oranges, lemon, watermelon, etc. are produced using this technique. So these are some of the examples of the fruits in which parthenocarpy technique could be adapted, and you can get the seedless fruit. This technique involves inducing fruit formation by the application of plant growth hormones such as auxin. So you remember this hormone name auxin. This is used in the process of parthenocarpy to develop the parthenocarpic fruit. Ideal candidate for the parthenocarpy in some of the example. Let us see that. Bananas. Reason, already naturally parthenocarpic in many varieties. You got this answer? Already the banana is producing naturally the parthenocarpic fruit, right? In many varieties. High commercial value. The banana is having the high commercial value, the parthenocarpic one. Continuous market demand. It is having a great demand, this banana, the parthenocarpic one. Seedless fruits are preferred by the consumers. Another example, watermelon. Reason, high market value for the seedless variety. So it will be good now. Watermelon without seed, it is easy to eat them, right? Otherwise, you have to remove all those seeds. Better eating quality without seeds. Consumer preference for the seedless types. And the good response to the growth regulators. Another fruit, cucumber. Why this cucumber is preferred for the to make the parthenocarpic fruit? Because they are readily response to the growth regulators. They are easily like responding to the growth regulators like auxin. Better fruit quality without seeds. High market value and improved processing quality. So these are the different reasons that we prefer to have a parthenocarpic cucumber. And beside these, we can grow the grapes, we can grow like pineapple, we can also have the oranges and papaya as a parthenocarpic fruit. So if there is no seed, then there is no ovule in the ovary. Yes, there will be no ovule. No, there are ovules. Listen, there are ovules, but the ovules do not transformed into seed because there is no fertilization. Parthenocarpic fruit, they do contain the ovule, but that do not transformed into the seed because there is no fertilization. So the remnants of the ovule will be there in the form of a sheath-like only. Like in the grapes, in the center, if you see, there are some sheath-like covering. That is the ovule, but that ovule do not transformed into the seed, right? Next question, explain the role of tapetum in the formation of pollen grain wall. Very, very important question. And this is from the section 1.2. So tapetum is the innermost layer of the microsporangium. No, they are not false fruit. They are not false fruit because still the ovary is forming the fruit. In the case of parthenocarpic also, which part is forming the fruit? The ovary. The false fruit is that fruit that the ovary is not forming the fruit, but any other part is forming the fruit, like receptacle or the thalamus, right? So that is the false fruit. The definition of false fruit is different. The floral part that is taking part in fruit formation other than ovary. But in the case of parthenocarpic fruit also, which part is forming the fruit? Still ovary only is forming the fruit, right? So parthenocarpic fruit is a true fruit. Ovary transforms into fruit, right? So false fruit is other thing. Okay, coming to the question, tapetum is the innermost layer of the microsporangium. So imagine the microsporangium structure. Outer wall is epidermis, then inner wall is endodermis, then there are several middle layers, and then the innermost layer is the tapetum. The innermost layer is the tapetum, right? So it provides nourishment to the developing pollen grain. You all know that this tapetum is providing nourishment to the pollen grains. During the microsporogenesis, the cells of the tapetum produce various enzymes, hormones, amino acid as a nutrients and other nutritious material required for the development of pollen grain, right? These are the things that are synthesized by this tapetum, and you all know that tapetum may contain more than one nucleus. They could contain two nucleus, three or four, like this, right? So it also produces the exine layer of the pollen grain, which is composed of the sporopollenin. Now see here, this is a very, very important point. So this sporopollenin, you all know that the exine is made up of the sporopollenin. So where this sporopollenin is coming, it is coming from the tapetum layer only. This tapetum layer is forming that particular substances, and that substance get deposit on the outer wall, and it forms the spore. That outer wall formation takes place, and that is by the sporopollenin, right? So sporopollenin is produced by the tapetal cell surrounding the developing pollen grain within the anther, right? Now the last question, what is apomixis and what is its importance? This question is from the section 1.5. Apomixis is the mechanism of seed production without involving the process of meiosis and syngamy. So here you can see no meiosis and no syngamy. No fertilization has been taken place, but still there is a formation of embryo. So that is the apomixis. So seed production is there. Apomixis is the mechanism of seed production without involving the process of meiosis and syngamy. It plays an important role in hybrid seed production. Okay, this play an important role in hybrid seed production. This we have discussed. The method of producing hybrid seed by the cultivation is very expensive for the farmers. And also by sowing the hybrid seed, it is difficult to maintain the hybrid character as a character segregate during the process of meiosis. So apomixis prevent the loss of a specific character in the hybrid. And also it is cost effective method for producing seeds. So once you develop the hybrid seed once a year, okay, it can be lost if you develop a new seed from that. So instead of forming like a new seed from that, you can go for the process of apomixis, right? So this is all about today's session. So thank you very much for attending today's session. We will meet tomorrow with a new session.